And it looks pretty much the same for all of us, except, look here. What I've highlighted for you there are what I'm going to talk to you about for the next 10 minutes. Those are called SNPs, single nucleotide polymorphisms, where we differ according to the genome, where we have one single nucleotide that's different, that there is a variation in the genes among different people. So that instead of having the same base pair in that sequence, some people have one base pair and some have another. These are mutations, just a single substitution. And how many of these are there in the human genome? Well, probably about 8 million, as best as we can tell, in the entire human genome. And this, to a large extent, is why we are not clones of each other. It's why we don't look alike. It's why we don't sound alike. It's why we have different personality characteristics. And it's why we have different vulnerabilities to diseases. So, let me be a little more concrete about it. And for those of you who are sophisticated in this area, I apologize, but I want to make the point one more time. So here are 3 individuals, and at the top of the slide is the triplet code. So you see these are the single nucleotide polymorphisms. These are the nucleotide base pairs, and the triplet code codes for an amino acid, alanine. Now notice in the second person, the adenine has been substituted with a guanidine, but it doesn't matter because both of these triplet codes code for the same amino acid, alanine. In that case, this SNP, the single nucleotide polymorphism, is considered non-functional. It makes the same protein. But now look at this person 3. In the second position here, there is a substitution. An adenine here replaces a guanidine, and in this case, the amino acid that's coded for changes from an arginine to a lysine. And now look what happens. Now we suddenly have a different protein. And this different protein has different abilities. It either is a different receptor, it no longer can bind the neurotransmitter as well, or a different transporter. It's a functional polymorphism. It hasn't changed wherever it's expressed. So now let's talk about how this might affect psychiatry. What you're looking at here is a polymorphism in the serotonin transporter. This is the protein that I showed you earlier that's sitting on the presynaptic terminal waiting for you to prescribe SSRIs. Right? So this is a protein that regulates the amount of serotonin in the synaptic cleft. And it occurs the gene is coded for by each of our parents. So one can have a long arm of the serotonin transporter, shown here with the arrow, or a short arm. And since you get one allele from each parent, you can either have an SS, 2 short arms, you could have an SL, a short arm and a long arm, or you could have 2 long arms, an LL. And it turns out that if you have the S allele, either as the homozygote or as the heterozygote, it is a functional SNP. You will make less of the serotonin transporter. And probably you will have fewer serotonin neurons. So you might ask, so what? Why is that so important? It's important because of this finding that was considered when it was published in the year 2003, the second most important finding in all of science. This was published in Science magazine, came from the Institute of Psychiatry in London, and Avram Caspi was the first author. What he did was he studied about 1,700 adults, age 18 to 26 in New Zealand. And he got information from them about their current mood state and also about child abuse that occurred between the ages of 3 and 11. And he genotyped these individuals and for the serotonin transporter. And what you see here on the y-axis here is the likelihood of having a major depressive episode, and on the